Hey everyone, sorry about that. Zoom made me update, of course. How's everyone doing? Welcome back. Feeling better? Thanks, Tala, for asking. Yeah. Still not 100%, I have to be honest, but I do feel better. And so happy to be back live. Hopefully you don't have to answer this, but I was just wondering like how the pre-recorded, I've done pre-recorded lectures in the past, usually for the really like some of the lectures are really complex and long, and so sometimes the pre-recorded version I feel is a better way to deal with some of those, because you can pause, as you as the student, you can pause and walk away and kind of like take a break if you need. So hopefully the pre-recorded version was okay. Luckily, like we have the discussion board thing, so that kind of helps to, what I recommend if we do have any more pre-recorded lectures, and we might have one or two for the rest of the semester, depending on things. But yeah, I recommend like while you watch them, you could even just be adding questions into the discussion threads in real time. Okay. So let's get started. I am going to share, let's see, our screen. Oh, okay, Melanie. And yes, good. Okay, good. People are trickling in. That's good. I wonder if other people had to update as well. Zoom seems to happen in batches like that. Yeah, it's weird. Like usually when I have to update, everyone else has to update too, although that's not always the case, but a lot of the time. Okay, today is not as much material as some of our previous lectures, partly because we're really starting to build now with what our vocabulary, what vocabulary we have and things like that. So I think today should feel pretty painless, even though there will be some new terms. Okay, so hopefully you guys can see the slides. Let me know if you cannot. So last time, well, let's first, last time, which was pre-recorded, we talked about the lycopods, right? We talked about Lycopodiophyta, and we talk about in particular Lycopodium and Selaginella, right? And then today we're gonna, and so one big new thing that we covered last class was this idea of homospory and heterospory, right? So plants with one type of spore versus plants with different types of spores. So today we're going to keep going with these spore shedding vascular plants and talk about the horsetails. We'll talk a little bit about the steel, not a lot, but a little bit. The evolution, the alternation of generations as it pertains to horsetails. And we'll touch on this idea of a megaphyll. So the evolution of megaphily is what it says here, but we'll talk about that briefly, but that will be something we get into more when we go into the ferns. So I like to get us oriented, right? We're in the spore, so we're up here in the vascular plants. We're up here in the spore shedding vascular plants. And so far we've covered the lycopods, and now we are going to get into those horsetails, and then next class we'll get into the ferns. At least that's the plan. Okay. Horsetails. Horsetails is a type of plant you've probably walked by it in your life. You may have even thought it was like a weird stick. But the phylum name is Equisetophyta. That's how you say that, Equisetophyta. But you can see them here. They're different looking. They don't look like a lot of the plants that you might be used to seeing. So we call them horsetails. Sometimes you also see them called scouring rushes. There's a ton of variability and diversity in the horsetails. They have kind of this general shape to them, which is kind of like a stalk, right, or a little stick. But there's a lot of variation. Some species have, here I will use blue. Some species have these kind of like whorled branches coming off of the sides. Others have kind of like maybe less of that. And some species have combinations of the two. So there's a lot of diversity if you look up close. Horsetails we call articulates because the stems are jointed. So this is how they might look if you see them in nature. Like I said, kind of like a weird stick. And if you were to go up to that weird stick, right, the horsetail, and kind of pull on it, it would kind of pop apart almost like Legos. It's like each of these little sections here, which we'll learn what they are in a second, but so like this little section here, this little section here, if you were to tug at this part here, the whole thing would like pop off, right? And that's what we see here. Here, the whole, oops, gotta mute. That whole section would just like pop off almost like a Lego. See, like, so if you were to pull on like this part right here, it would just like pop off right at that spot there, like a Lego. So for that, we call them articulates. That's why we call them that. Henry says, yeah, yep, like markers. Yeah, it's weird how they do this. It's most likely the result of living in an aquatic environment. Probably also a way to deal with potential herbivory, although there's reasons why, meaning getting eaten by something. But there's reasons why things would probably not want to eat this, which we'll talk about in a sec. Okay, so horsetails. When you look at this, right, when you look at this picture, you probably don't think of a horse's tail, right? Fair enough, I get it. But that is what the name means, or where the name Equisetum comes from. So, like I said, you might have to have a little bit of a stretch of the imagination depending on what species you're looking at. But equus is the Latin word for horse, and then seta is the Latin word for hare. So Equisetum, right? That's where it comes from, means the horse-haired phylum. Paula says she loves horses. But yeah, so you might not think it like I said, look, if you look here, these are different animals in the genus Equus, right? Because remember, the animal kingdom also has genera, right? Like Homo sapiens, right? So all of these animals shown here, oh, cute little animals, right? Fuzzy, they have eyes, are in this genus Equus, right? So zebra, horse, etc. And like, yeah, a horsetail, I don't really see it, but okay, fine, right? If we like kind of look at it from a broad perspective, sure, right? Like I can see where somebody may think that. And as we see on subsequent slides, there are some species that definitely have more of a horsetail-like vibe than others. Just like the lycopods we looked at last time, the horsetails are super old. They also arose around 400 million years ago. They were also dominant members in those carboniferous forests we talked about, right? Those old swamp forests when the continents were pushed together with a lot of land mass around the equator, creating the ability to have these kind of very shallow swamp forests. So horsetails were also very significant for us in the formation of coal. And on the bottom here, you're seeing a reconstruction of a horsetail forest on the left bottom. And on the right, you're actually seeing a fossilized horsetail. Some general characteristics. Well, we're in the vascular plants now, so we know the Sporophyte generation is dominant, right? So when we're looking at horsetails, we're looking at the Sporophyte generation. To make things easier for us, there is only one genus in horsetails, which is Equisetum, so that's nice and convenient. There's about 20 species in that genus, and some of them are occur naturally in New Jersey. So that's why I said there's a really good chance that you've probably come across these during your life in New York, New Jersey area. Two species that are found in New Jersey are Equisetum hylamale, so that's this one. And so the way you say this is hyamale, like hyamale, that kind of thing. Equisetum hyamale, which is hi, shown here on this slide, right? So Equisetum hylamale is one of the ones I would say does not look like a horse's tail, right? This does not, to me, look like a horse's tail, right? This does not, to me, look like a horse's tail. But if we were to look at, yeah, I mean, it definitely doesn't look like a horse's tail. But so Equisetum hyamale is one that you would maybe see in New Jersey. And then the other one you might see in New Jersey is Equisetum arvenzie. Remember, with this E here at the beginning, that just is referring to the genus name, right? We already learned that. So the previous slide showed Equisetum hyamale, and then this slide is showing you Equisetum arvenzie. That's how you say that, arvenzie. And Equisetum arvenzie, fine, looks more like a horse's tail, right? Like this looks more like a horse's tail. If we were to look at a whole bunch of them in nature, shown here on the bottom left, right? That has a bushy appearance, looks like a bushy horse's tail, fine. I would still argue if your horse's tail looks like this, probably sick. Like, get it to a vet. But fine. All right, where do you see them? So first I should say there's no significant economic importance. We don't really make money off of horsetails. In fact, sometimes we spend money to kill horsetail plants because they can cause death to livestock. If you're raising cattle or something like that, and they eat a lot of the horsetails, it can cause them to get sick and die. That's because inside of the plant is a chemical called silica, which is not good to eat. But where do you find them? So a lot of times near water is where you find them. So kind of like in sandy environments on the land, you'll find them. You could also find them growing, emerging, or having an emergent habit coming out of in aquatic or water environments. If something is emergent, so if you see horsetails, maybe I should change colors because this is kind of, let's do red. So you might see them growing on the land in sandy environments, or you may see them growing in aquatic environments as an emergent species. What does that mean? Emergent means it's not fully under the water, but it's not floating on the water. So it's growing at the bottom of so let's say it's a pond, right? It's growing in the pond. It's attached to the ground underwater, right? It's attached. But the majority of the plant comes above the water, right? So that's what we call emergent. So emergent example shown here on the left, terrestrial kind of sandy environment shown here on the right. Also shown here on the right is that they can often occur in disturbed areas. So for example, along this railroad track where there has been, they had to build the railroad and it's pretty, stuff like that. So Henry's asking, what is kelp? Kelp then, kelp is attached to the ground under the sea. And so it's a submerged. It may appear floating toward the surface of the water. But anyway, so yeah, so horsetails, disturbed areas. So that's another reason why you've probably come across them, because a lot of Newark, New Jersey and New York City in general area, super disturbed, which is a habitat that they can live in. All right. We're in the vascular plant, so we know the sporophyte generation is dominant. We also know that the sporophyte generation is vascular, right? Duh, because we're in the vascular plants. And we also know that the sporophyte generation has true roots, true stems, and leaves. So we know that. Some building here from terms we learned last time. So in all the members of Equisetum, right, in all 20 species in the genus Equisetum of the horsetails, there's one type of sporangium. So there's homosporous sporophyte plants. One type of sporangium, they make one type of spore. So we can say, I'll use a different color here. So we can say that the sporophyte plants are homosporous, right? Same spore, right? Homo, same, spora, spore. So the sporophyte generation is homosporous in that it produces one type of spore. And we can say that the gametophyte generation that will grow out of that spore will grow exosporically, right? The gametophyte generation is exosporic, meaning growing outside of the spore. If these terms seem confusing or you're like, I don't know what she's talking about, go back to the last lecture where we covered this a lot. Okay, how does the sporophyte generation grow? Oh, our good friend apical meristem is back, right? Apical meristem. So on the tips of the roots and the tips of the stems will be this apical meristematic cell. If you look at the tip of a stem, for example, under the microscope slide, which is what we see here, the apical meristematic cell is very easy to identify, or easier to identify maybe than other cells, because it has this really nice three-sided, almost like a triangle cell. So here is where it's located. The contrast on this image here is not super great, but trust me, when you're looking under the microscope slide, it's very noticeable because it's usually large and shaped kind of like an upside down pyramid. I think I highlight that. Yeah. If we were to put like a drawing on top of this microscope slide, my head is probably blocking it, but what that says is apical meristem cell is shown in those that dotted area. And something we'll learn about, I think, a little later, maybe not today. I'll change colors. As that, remember the apical meristem, right? The cell there is like a stem cell, right? Apical meristem, right? Stem cell. So as it divides and creates new cells, the cells it creates can be root tissue or parts of the stem tissue or even leaf tissue. And so that's what's showing you here. So as this cell, as this apical meristem cell divides and divides, this area here is now on its way to becoming new stem tissue. Back here, when the apical meristem cell divided and left that material back there, that's going to grow into the leaves. Yeah. Okay. Still talking about the sporophyte generation here. So we know it grows to these apical meristems. If we were to look at a sporophyte, you'll notice this like very, most of the plant is this, what it says here, horizontal rhizome. Remember, rhizome means horizontal stem, right? Horizontal stem. So if you look at a horsetail, most of its body will be like a stem like this that's growing horizontally, similar to what we saw in lycopods. And then occasionally you'll have these vertical stems come out. And where those vertical stems will come out is where the meristem cell left those cells that will become stems. So I'm just trying to get you to think about, like, if you think about even a human being, right? And you start with like not that many cells, right? A brand new human being starts with not that many cells. And then there's more cells and more cells and more cells. All of those cells going on to become parts of the human body, right? These cells will make eyes, these cells will make, etc., right? That happens in plants too. You don't just go like, oh, here's a baby plant and then whammo, a plant. It has to grow. And so the new leaves, go back here for a second. As the apical meristem cell divide, it will create leaves, which will show up at these leaf primordia. Leaf primordium is singular. But as it grows, it will also leave behind, oops, shoot primordium, which is where the stems will grow. Okay, enough of that. We'll discuss that more later. Oh, this is a new thing for us, is this concept of having dimorphism. So just to get you not scared, what's a good color? I guess like blue is good. So you see here, some species have dimorphic upright branches. Di means two, which we've already covered. We've already come across di when we learned dioecious, right? So some species have dimorphic branches. Di means two. Morph means shapes. Two different types of shapes. That's all dimorphic means. What do we mean by two different shapes of branches? Well, Katrina, you have a question? Here, I'm going to mute you. What do we mean by two different shapes? We mean, well, let me move this. That there's some, okay, so this right here on the left is one single species of horsetail. Some of the upright branches or the shoots are sterile, meaning they don't make any spores. And in this species here shown, that's this fluffier, horsetail-y looking branch. But other branches on the same plant might be fertile, sometimes green, sometimes not, and have on the top the strobilus where all the sporangia are located, right? So that's what we mean by dimorphic branches. Two different types of branches on the same plant. One other important thing to mention about the horsetails is for all members, for all the species of horsetails, it is the stems that are photosynthetic, not the leaves. So that's why the stems usually have this kind of green appearance. So only the stems are photosynthetic in horsetails, not the leaves. And the stems are divided into these things that are called nodes and internodes. I guess yellow, no, I guess black, which is what we talked about before with like the Lego kind of action. So like if you were to pull on the stem, they would pop apart at the nodes. That's why we call them articulates here. So like in this here, right, this area here is a node, this area here is a node, and inter means between. So the internode is the area between the nodes. Node and internode are areas or regions. They're not, so there might be different, depending on what plant you're talking about, different things might occur at the node, right? Like in some plants, you might have leaves occurring at the node, or maybe a branch occurring at the node. So I just want you to know that node and internode are locations, right? Like, oh, where's the movie playing? Oh, it's playing at the movie theater. The movie theater is the location, not the movie, right? It's the location. Michelle's asking, do bamboo have nodes since they look similar? Yeah, so basically pretty much every plant we're going to look at, well, many plants that we're going to cover in this class going forward have nodes and internodes, which is why there's a lot of building in these lectures, right? So we'll see nodes and internodes in many things, and yes, bamboo does have them, but it's a little more complicated in bamboo because bamboo is technically a grass. So it's a little different, but sure. Yeah, for our purposes today, sure. Yep, but yeah, we'll see nodes and internodes a lot. So that I show here the nodes, and then look, there's the area between the nodes, which is the internode. So again, nodes and internodes are a location on the plant, not a thing per se, not a thing, right? They're a location. What else about the Sporophyte generation? Okay, so the stems, if you were to look at the horsetail stems, Halva says, wow, didn't know bamboo is a grass. Yep, it is. That's why it's a little different than this here, but good way to think about it. If we were to look at the stems up close, you would see this kind of ribbing on them, which is what's trying to be shown, I guess, yellow. You might see it here. It's kind of like these vertical lines. If you were to rub your finger on the sides of the plant, you would feel that those are raised, right? Those are like ribbing. What do? They serve to help support the stem. So they provide like structural support. You can think of it almost like a scaffolding, makes it harder to break. And then inside the stem, as we mentioned already, in those, we didn't say they were in the epidermal cells, but horsetails contain silica, which is kind of gritty, and those can be found in the epidermal cells. So that's why before I was saying, like, these things like ribbed stem and silica in the cells makes these not really great to eat. They honestly, if you ever touch them, they honestly feel like a piece of plastic. They don't feel like a living plant. They feel man-made. That's because we make stuff out of silica. And no, not like a cucumber. It literally feels like a piece of plastic. And see, so speaking of that, this is something people used to do before we made things, right, out of before we made things like steel wool. So part of the reason, so we call horsetails because they look like horsetails, but we also call them scouring rush, right? And so part of the reason we call them that is because people used to use these plants to clean pans. So I don't know if you've ever, yeah, Nan's like, do they feel like dollar store plants? Yeah, like kind of like that, like a plastic thing. But so back in the day, this is something people used to do. So I don't know if you've ever cooked, maybe, let's say maybe you cooked rice or something that had to be kind of going for a minute, right? And you got distracted because you were listening to my awesome lectures, and oh no, like this happened to your pot, right? I don't know why you would be cooking with this on. I don't know, maybe this person's cooking something suspicious. But anyways, the point is, like, this happens, right? This happens. And when that happens, what do you do with it? So nowadays we do something like this, right? Maybe this is why this person has the gloves on, because they're in the middle of trying to like deal with this disaster here. Nowadays we use something like steel wool or something like that. But back in the day, before people had things like that, people used horsetails, and you can still use horsetails for this purpose if you so please. If you want to be greener and not have to deal with things like sponges and stuff, go get yourself some horsetails and you can clean your pots. But again, this is because of the things I just mentioned on the previous slide, that These things they almost feel like a piece of plastic, and they're super strong. And so that allows you to kind of scrape your pan. All right. I mentioned they were I mentioned they were a big component. Yes. So if you, Mara, yes. So if you touch them, they feel rough and like a piece of plastic. So I mentioned that these plants were, or members of the Equisetum phyta, were part of those carboniferous swamps. And those members of Equisetum phyta, those are not called Equisetum. They're called calamites. And so they look a lot like modern day horsetails. They're now extinct. They were a lot bigger. If you go back to that slide that mentions the Carboniferous forest, and I showed you that reproduction of what, like, oh, here's a horsetail forest. It was showing you these things. So they're extinct now, very similar to modern day horsetails. One main difference though is that they're they were super big and tall, much bigger and taller, almost tree-like than those. But we see some of the same things I just mentioned, like here's that horizontal rhizome, right, which would continue. Here's the vertical part of the stem. This one looks kind of like a horsetail, right? It's fluffy. That also has nodes and internodes. And actually here on the right is a fossil of calamites, which we should have in the lab. So next lab, you're going to look at the lycopods and horsetails together, which should be fine because I don't think there's that much material for the horsetails. But hopefully one thing we have that day is some horsetail fossils, because then you'll be able to really see the ribbing, like vertical ribbing that I mentioned. And then here's a node. Here's a node. And then this section here would be the internode. So some of the same stuff that we just covered. So Hal was asking about if the silica is bad for humans. Yeah, you don't want to eat horsetail. Don't eat horsetail, but you can use it to clean, say, a pot. But yeah, just the reason why it's bad for animals is because the animals eat it. Yeah. I don't know if you guys have ever gotten, I think it's the same stuff. Have you ever bought a pair of new shoes or like a new jacket or something, and there's those little packets inside that say, don't eat this? I'm pretty sure that's also silica. Yeah, just not something good to eat. All right, so yeah, you'll see fossils. Now we're going to talk about, yeah, okay, thank you, Kira. Yeah, that's what I'm saying. Like it even says on those little packets, don't eat this, right? That's because it's not good for you to eat. All right, before we move on, we got to talk just a little bit about the steel. Yeah, don't eat it. Which, as we learned before, recall from lecture 4, the steel is a way that we describe how the xylem and phloem are organized within a plant, right? Within a plant. We learned already the Lycopodium had the protosteel. Do I do that again? Yeah. We learned that Lycopodium and those earlier vascular plants, based on the fossil record, had protosteels, right? Kind of a solid column of xylem and phloem tissue. The slightly more or later evolved plants like Selaginella have that saphanosteel with no leaf gap. So here, just as a refresh, right? Protosteel, the way you can think of a protosteel is just like a solid, like this whole thing is, if you were doing a cross section, a solid column, right? Solid. Saphanosteel with leaf gaps, sorry, with no leaf gaps. So protosteel is what you would have in, in Lycopodium. Just a solid column, solid tube of xylem and phloem. Saphanosteel with no leaf gaps has like a ring. a ring of xylem and phloem tissue with a pith in the middle and no leaf gaps. If you get to plants we haven't covered yet, we'll get to what's siphonosteal with leaf gaps, which looks similar. You have the same ring shape, but occasionally it might look like this instead, because there will be this leaf gap, which we have not covered yet. And then eustele is kind of like tubes. So it no longer kind of centralized or in a ring, but basically like little, so if you did a cross section, it would kind of look like this, like little tubes, the tops of like tubes. So that's a good way to draw those out to kind of keep them in your brain. But anyways, so Lycopodium had protosteel. Selaginella had siphonosteal with no leaf gaps, right? This is showing here protosteel. Equisetum has a funky steel. It sounds like a song, like funky steel. Equisetum has a funky steel in that it's kind of a combo of a eustele and a siphonosteal. So what do I mean like by that? Remember here I said a eustele is like little straws. You can think of it as like straws. So eustele is kind of like little straws that are not necessarily arranged in a ring or anything like that. Siphonosteal, which is the two center ones, is like a ring, right? And so Equisetum is kind of a combo because it's like a ring. It's like the straws are arranged in a ring. So this is a cross section through horsetail. Notice the vertical ribbing. Look, whoa, right? Whoops. Notice the vertical ribbing. That's what I was saying. If you rub your fingers on it, you feel those little bumps. I think I probably, let's see. I just want to see what okay. So I just want to, sorry, I'm not trying to make you dizzy. I was just seeing if I have to draw it or not. All right, I guess I do. I guess I should point it out. So it's a eustele like siphonosteal, right? That kind of sounds weird too. Like, you still like siphonosteal? What is wrong with you? So you still like siphonosteal. Siphonosteal means in a ring. Eustele means like straws. So the vascular tissue in horsetails are like kind of like straws or a eustele, but the straws are arranged in a ring. So I'll use red, and let's get rid of this here. I'll use red. So you see here, vascular strand. Vascular strand is just a fancy way of saying steel. Or sorry, saying vascular tissue. So xylem and phloem. So here is one area of xylem and phloem. Here's another area of xylem and phloem. Here's another area. So these are those straws that I'm talking about. Xylem and phloem, xylem and phloem. So these are the eustele part because they're in straw shape, but they're also arranged in a ring. And that's the siphonosteal part. So that's what that means. You still like siphonosteal. All right. Epidermis, we've covered already. In the horsetails, that's where the silica is going to be, that we already covered. The stomatal apparatuses will also be in the epidermis. These circles are showing you where some of those are. You have the cortex. Remember, that's the ground tissue in the stem. Go back to I think it's lecture 4 to get refreshed about the different types of tissues. In the cortex, you have, okay, don't get scared, sclerenchyma tissue, which is a fancy way of saying fiber cells. You have parenchyma tissue, which is the most common cell type in plants. You have molecular canals, and you have an endodermis. So what are those things, Rebecca? I'll show you. So here's the cortex. We're doing close-up of the cortex, right? Here are your sclerenchyma or fiber cells. They usually stain very dark. You could see them kind of close to the outside of the stem, right? Their job is mechanical support, so that makes sense. There is those parenchyma cells. They usually stain kind of bluish green because they're full of chlorophyll because they are the area of photosynthesis. Then if you would see, so if you're seeing here, if we go back here, you see kind of a bunch of holes going on, right? You have like these larger holes and then the smaller holes. The smaller holes are those vascular bundles, right? So like xylem and phloem, etc. The larger holes are the vallecular canals. So if we zoom back into our slide, we're pointing here now to those larger holes. And what the vallecular canals do is remember, horsetails are growing with the bottom of their, with some of their plant body growing underwater and some of their plant body growing above the water, right? Well, if you're growing under the water at all, gas exchange is going to make it be a little difficult, right? Because you're growing underwater. So these vallecular canals run the length of the vertical shoots and allow basically air to be inside of the plant. It helps with that. The reason they are called vallecular, I guess we'll use blue. Look at this. Look, I'm going to draw up here. Have you ever heard of like valleys, like peaks and valleys? So you can think of the outside of the stem as being like a peak and a valley and a peak and a valley. And so the vallecular valley, right? Vallecular canals occur in the valleys, right? That's why it's called that. Help with gas exchange. There they are, circled for you. Then the endodermis, this is a layer of cells, kind of sounds like epidermis, right? It's similar in that it helps protect the plant. So the epidermis is like the outside, so it's preventing things from coming from the outside of the plant into the plant with its cuticle. The endodermis is further inside, right, below the sclerenchyma tissue, below the parenchyma tissue, closer to the vascular bundle, which is here, right? This is where the xylem and phloem is located. And basically this endodermis, which is kind of, for what it's worth, it's kind of hard to see in this slide, but it's kind of like this area. It's kind of hard to see. But its job is to keep things from going into the xylem and phloem tissue. So it's an extra layer of protection, keeping things like bacteria and fungi from going into the xylem and the phloem. That's really useful because your xylem and phloem are moving water and food all throughout the plant body. It's like our vascular system, right? So if we don't want a bacteria or a fungus to enter our veins, because then the bacteria or fungus can go all over our body. So same with plants, the endodermis helps prevent things from entering the vascular system. All right. This is what I was thinking with here, right? This is back to that you still like siphonosteel thing. You still like siphonosteel? In that you have these vascular bundles that are arranged in vascular tissue, vascular bundles that are arranged in these vertical rods, or you can think of straws, and the straws are in a ring. That's what I drew back there. But if we're going to look at each vascular area up close, right? Here's one vascular strand. You'll see the vascular bundles of xylem and phloem and a corinal canal. So basically, the easiest way to spot, remember before, here, I'm getting distracted. Remember before I said there's two holes, there's two hole types, right? There's these larger ones, which are the vallecular canals. And then there's these smaller ones, which is where the xylem and phloem are located, those smaller holes. Those smaller holes are called, I'll use a different color, black. No, I'll use yellow. Nope. Yellow is corinal canals, right? So here I'm drawing in green is a corinal canal. And then around that, I guess we'll use, you'll first find xylem tissue, which in this slide looks like that. And then phloem tissue. And that will occur at each of those locations. There's a question, how can you tell the difference between phloem and xylem? So this slide is from your book. This slide is from your book, and it doesn't do a good job of showing you this, but xylem tissue and phloem tissue always stain different colors. So that's one way. Usually xylem tissue stains kind of a red color. It depends on what you're looking at. So they will stain different colors, and also xylem tissue is always located closer to the center of the, I mean, this will depend on what in particular you're looking at, but in terms of this here, xylem tissue always occurs closer to the center of the stem. So if you find a corinal canal, right, what did I use, green? So if you find one of these corinal canals, the xylem tissue, what did I use, black, will always be the first layer here because it's closer to the inside of the stem. And the phloem tissue will always be kind of next to that toward the outside of the stem. So Natasha says, so the phloem will always be on top of the xylem. Well, it depends on where you're looking. So if we go here, oops. If we go back here to not being so zoomed in, it will depend because you got to think of the center of the stem. So if we were to take, let's take this one here. Here's a corinal canal, right? I don't know if you're seeing me draw it there in green. And the center of the stem is here, right? So the if you look at that corinal canal, there's the xylem, and the phloem will be here. So in this case, from this orientation, yes, the phloem is on top of the xylem. But if we were to look at the corinal canal here, down at the bottom, the xylem tissue is always going to occur closer to the center, which is here. So if we look at this one, here is the xylem tissue, and phloem tissue would be there. So in this orientation, the xylem tissue looks on top of the phloem tissue. That's why I'm saying focus on the center of the stem as versus the outside of the stem, right? Xylem tissue always occurs closer toward the center of the stem. Yeah, Kira's asking, is that why last lab some of the stains were red? Some of the slides were red and some were blue. Yeah, so different cells stain different colors when you prepare microscope slides. Xylem tissue, we haven't covered this yet, but xylem tissue is always dead when the plant is alive. Xylem tissue is dead. So, and phloem tissue is alive. So when you cut off a piece of plant to make a microscope slide and you give stain to the microscope slide to make it look better, for you to see things better, the dead tissue and living tissue stain different colors. And in addition to that, sometimes the components of the cells will stay as well. And yeah. All right, we gotta move on. All right, Henry, I can't talk about that today. We will talk about that another time. All right, so we went through this corinal canal. We're not really sure what corinal canal does. We just know it's there, and it's an easy way to find the xylem and the phloem, because if you see this little hole, we know the xylem and phloem are located very close to there. We think maybe it helps with water transport. Remember, those vallecular canals that are super big help with keeping air inside of the stem of the plant. Corinal canal, we're not sure. We think it's to help with water. Then in the middle, there's this pith region. That's this giant hole. I'll do a yellow here. So this whole area is the pith region. It's not, yeah. So it's basically this large hole in the center of the horsetail, and it's surrounded out here. I'll use green. All this here, all this here, all this here, etc., is that parenchyma tissue. You'll even find some of it here. Parenchyma is the most common cell type in plants. All right. Oh, and the reason why I'm showing you this, so this is a modern day horsetail. This is a fossil horsetail from Calamites. And look, what do you know? Same things we just talked about. Look, here's the pith region in the middle. Harder to see here. Harder to see here, but there are, I'm assuming, because it's harder to see here, these larger holes that we see are those vallecular canals. And there are probably smaller holes next to them that are hard to see on this slide. But either way, the point is it's still a eustele like siphonostele, right? You still have the vascular bundles arranged in these kind of straw shapes. Xylem and phloem, xylem and phloem, xylem and phloem, xylem and phloem in like these little straws. And those straws are arranged in a ring. So even extinct Calamites had the eustele like siphonostele. Okay. Horsetails have branches, something we haven't really covered yet because none of the plants we've looked at really today have really had something we can call branches. But I did mention at the top of the class, these branches will come out of those shoot primordia. That's located in the cortex. The shoot primordia, you know, I don't care that you know this, so we'll move on. Node and internode. Just as a reminder, branches will come at the node. So remember, I said node is a location, right? Not a thing, a location. So like, where are your friends? Oh, your friends are at the grocery store. The grocery store is a location, not your friends. Your friends are not a grocery store. The grocery store is a location. So where are the branches? Oh, the branches are at the node, right? The branches are at the node. The node is a location, not a thing. Not all species of horsetails are branched, but if they are, if the species is branched, there will be a branch for every leaf. And the branches are in a arrangement that we call world, not like Earth, not like the world. World meaning kind of in a spiral, I guess is the best way. So like on this slide here, here's a branch, here's a branch, here's a branch, here's a branch. Notice they kind of go all the way, well, not here because they removed it for the purposes of showing you this, but they go all the way around, right? All the way around. And that is in a shape we call world. And here it says, I'll use red. Here it says there is a branch for every leaf. And then I'm showing you this and you're thinking, there's no leaves. I don't see a leaf. So what is she talking about? There's a branch for every leaf. There's no leaves there. Yes, there are. They just don't look like leaves that you're used to seeing. So on this previous slide here, believe it or not, these dark things These dark things here are actually the leaves. The branches have not yet emerged from these leaves on the top, but they are emerging from the leaves at the bottom. But the leaves the leaves in horsetails don't look like leaves that you're used to seeing. They are very, very simple and small. They also have one vein. They are also whorled at the node, meaning going around. So I guess I'll use yellow here. So this one thing is a leaf. Next to that is a leaf. Next to that is a leaf. Yes, trust me, these are leaves. Here's a leaf, here's a leaf. And if we were to look closer up in a different species, this little thing here, that's one leaf. Here's a leaf, here's a leaf. So super, super simple, super, super small. They do have one vein and they are simple and they are very small. So you might be thinking, if you've been keeping up with the class, well, that sounds a lot like a microphyll, but it's not. It's not. And I'll tell you why in a second. So you'll find about 10 to 30 leaves at each node. It's like here we see what, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 just on this side, right? So there's probably about 30 at that node. Most importantly though, make a note, non-photosynthetic, non-photosynthetic. Why? Well, first of all, what part of the horsetails are photosynthetic? The stems, right? The stems are photosynthetic. Look how green stem. So that, for that reason, they don't need leaves to be photosynthetic. Also, keep in mind, these are growing in water. And so maybe having big photosynthetic leaves didn't work so great. But having photosynthetic stems was a better way to deal with living in that type of environment. Yeah. So this is what I was saying on the previous slide, and we're finishing up here, right? So despite the fact that these are very small leaves and very simple, and they have one vein, in the horsetails, these leaves are not microphylls. They're not microphylls. The reason they're not is because they have a different evolutionary path than the microphylls. You might recall, if you were awake that lecture, that when we talked about the microphylls, we think came from enations, right? Go back to the slides on enation theory. We think that microphylls came from enations. Larger leaves with multiple veins did not come from enations. And we haven't covered megaphylls yet. We will cover megaphylls, I think, in the fern lecture. But trust me, in the horsetails, when we look at these leaves, the way we got to these leaves here were by the ancestral types of horsetails having megaphylls, which we'll learn about in another class. But the megaphylls, because of living in these aquatic environments, have become highly, highly reduced. Oops, highly reduced. So to show you, for example, here's another example of an extinct horsetail, right? This is Sphenophyllum, which I think we'll also see fossils of in the lab. And look at these leaves. So these leaves look more like the leaves you're used to seeing, right? This right here, this is 3 leaves. These are 3 megaphylls. We'll learn about megaphylls in another class. So the ancestors of horsetails had megaphylls, but the modern day horsetails, those megaphylls have gotten super, super reduced over evolutionary time to look like this. And yes, they look a lot like a microphyll, but they're not. They're not. Roots, they have true roots. The roots come out of the rhizome. Remember, rhizome is a word that means horizontal stem. We talked about adventitious roots before in lycopods, but basically roots that just arise wherever the stem touches the ground. And the roots will come out of, really care that you know where the roots come out of, but they come out of the root primordia from the cortex. All right, now we're just going to talk a little bit about the gametophyte generation. We'll talk a little bit about the alternation of generations, and then we will be done for the day. So at the top of the class, I said horsetails have homosporous sporophytes and exosporic gametophytes. If these terms seem scary and confusing to you, go back to the previous lecture where we cover homosporous and heterosporousness, and go back to there also to refresh yourself about endosporic or exosporic gametophyte plants. But since we already covered it, so horsetails have homosporous sporophyte plants, and they make sporophyte plants make one type of spore. The spore that they make will land on the ground and germinate into a gametophyte plant, and those gametophyte plants will grow outside of the spore, like we're used to dealing with in all the other plants we've dealt with except Selaginella. And those gametophyte plants will be monoecious in that they have one house, one plant with both sex organs on them. So here we go. This is a gametophyte plant of horsetail. Notice the female and male sex organs there. This is what I was just saying. Very simple, phallus-shaped gametophyte plant, photosynthetic gametophyte plant, grows outside of the spore, right? It's exosporic. Monoecious or bisexual in that it has both male and female sex organs on one gametophyte plant. You should get used to the idea though that there are always exceptions in plants. There's so many plants that there's always going to be an exception to the rule. So yes, most of the time the gametophyte plants will be bisexual, but they sometimes can just be what's called like functionally unisexual, in that maybe a gametophyte plant is capable of making both sex organs, but maybe the sex organs are mature at different times, right? One reason you might want to do this is because then you're not fertilizing yourself, right? So it's beneficial to have sperm and egg come from two different individuals. So to prevent self-fertilization, you might stagger when your sex organs are mature. And yeah, some species just, some are messed and they just have antheridia. So always exceptions to the rules. All right, go through alternation of generation. We don't have to go super slow because we've covered most of these terms, but I will spend more time on stuff that's new. This is from your book. Here you go. You have a sporophyte plant, mature sporophyte plant, right? That's diploid. Look at all the structures on it that we've already covered here. I'm going to change colors to blue, right? Look, node, right? Here's a node. Here's another node. The area between the nodes is the internode, right? Inter, it means between the nodes. Here's our horizontal stem or our rhizome. Here are those weird small leaves that occur where? At the node. And we talked about branch dimorphism, right? In that some species have branches that are sterile and look kind of, in this case, like a horse's tail. And on that same plant individual, you'll have other branches that are fertile in that they make spores. So this is showing us a good example of branch dimorphism. If we were to zoom in, so we're going to zoom into the strobilus now, right? Here's the strobilus. We're going to zoom in and look at what's going on up there. So strobilus we're used to from the horsetails, right? We learned strobilus already. It's basically a way to arrange sporangia, right? But horsetails have one little extra component that we haven't covered yet, which is these things called sporangiophores. Sporangiophores, and my head might be covering it. But basically, on an old strobilus that we looked at before, you would just have like, here, I'll draw on the side real quick. I'll use this. So in the strobilus that we used to look at in Selaginella and Lycopodium, you'd have a stem, and then, where's my colors here? Then you would have, on that stem, you would have leaves, right? Leaves that have sporangia on them, or sporophylls, right? This is the old strobilus from Lycopodium and Selaginella. And so those leaves had sporangia on them. We'll use pink, right? So here you go. There's a sporangium. There's a sporangium. This is the strobilus we're used to looking at. In horsetails, there's an extra structure. I'll draw it here on the right. So we still have a stem, but instead of leaves now, I'll use red since that's what I colored it. We have these new structures that are called sporangiophores that kind of look like a mushroom. So this is one sporangiophore. In cross section, kind of look like a mushroom. There's another sporangiophore, another sporangiophore. Oh my gosh, and here's another one on the other side, right? Sporangiophore is going to have on it sporangia. So here's sporangia on one sporangiophore. The benefit is that, look, in the previous strobilus, you could only have one sporangium per leaf. But what's cool about the sporangiophores, little mushroom looking things, is you could have so many sporangia per sporangiophore. For example, so here I'll draw it in red. So, or I'll use the same, white's not going to be good, but here's the stem, right? In the middle of the strobilus. And then each of these mushroom looking things, here's one, here's two. Those are the sporangiophores, right? Here's another one. Here's another one. And then here is one that's been pulled off. But notice when you look at it up close, each sporangiophore is able to have way more than one sporangium. So this one has 1, 2, 3, 4, 5, 6, 7. There's, on this example, there's 7 sporangia on one sporangiophore. So that's way better than one sporangia per leaf. I gotta move on from sporangiophore. Yeah. But suffice it to say, strobilus still is a thing that has the sporangia organized. In the case of horsetails, they just happen to be organized on sporangiophores instead of sporophylls. If we were to look at the sporangiophore up close, just to mention, right? Here is one sporangium, right? On this strobilus, that's where meiosis is going to occur. And you go from the 2N spore mother cells to haploid spores. This is just to show you what sporangiophores look like in real life as versus a drawing. So remember the word for means to bear or carry. Sporangio refers to sporangia. So sporangiophores are things, structures that bear or carry sporangia. And just to give you the same kind of idea, like this here is showing 2 sporangiophores. They kind of look like little mushrooms. And look, 1, 2, 3, 4, 5, 6, 7, 8, 9. Like each of these sporangiophores has on it like 20 sporangia. So that's way better than 1. That's why this occurred. More spores. Eventually the spores will be released from the sporangia. Horsetails also have elaters, which we encountered before, which are basically little wiggly things that help the spores get released. Although in horsetails, what's cool is the elaters actually kind of wrap around the spores. So this is showing you a microscope slide of horsetail spores. And each of the spores are kind of like wrapped up in these weird elaters that will kind of move around and shake to help the spores get released. Eventually the spores will land in the ground, grow into the young gametophyte plants. Gametophyte plants have on them, we already know this, sex organs, archegonia and antheridia, egg in the archegonium, and sperm from an antheridium will eventually fuse via fertilization. And then you have the beginning of your new Sporophyte generation, which leaves us here. And I think, oh yeah, and the last thing is just, so that's it for alternation of generations. Most of the players are the same, right? There's a few new terms, sporangiophore being one of them, and node and internode being maybe new ones, and branch dimorphism, vegetative shoot, fertile shoot, fine, those are new, but everything else here we've pretty much already covered. Yeah, so some people have used this Equisetum arvenzie, which occurs in our region, as like an herbal remedy to treat certain things like bleeding, tuberculosis, even kidney problems. So it'll show up sometimes in classical paintings or writings about being something to use for these treatments. But I would just caution that you don't just go out and start like healing wounds with horsetail. I think there should be more studies done to figure out what the effects are. There's one study here, but maybe this is something that somebody could look into one day, if this is a good thing we should use, since it was used in other in the past. All right. Right before we end, I should just mention, so assignment 3, which has been posted since last week, since last Thursday, I believe, yes, is due this Thursday before 10 a.m. It covers the introduction to vascular plants and then the lycopods. And just a reminder about my late policy. And next class, we'll talk about the ferns, which honestly, I have to be honest, I love ferns so much. So the next class, and I think the class after that, should be a lot of fun. They're, in my opinion, way more charismatic than the horsetails and things. But yeah, so if anyone has any questions, I'm happy to answer them here. Otherwise, I will see you guys on Thursday. And I'll post all this material later. Thank you guys. If anyone has any questions. Yeah, have a great day. All right. I'll wait a second in case somebody's typing. You could also feel free to just unmute if you do have any questions. Thank you, Kiana. All right, well, if you do have any questions and you want answers to them, send me an email. We can also meet for office hours if you would like to do so as well. So have a good one, guys.